So I'm Mark Warren, Senior Director of Transporter Services, overseeing the transporter assays that we do in our Santa Clara, California lab. And I'm going to be talking today about a project that's a little different than most of the projects we normally do. So I'm going to be talking about the DDI potential, not necessarily of drugs, but of molecular excipients. So I'll start out just my introduction here, my outline of my talk. One of the things I want to mention, drugs are more than just an active pharmaceutical ingredient, which is something we don't appreciate all that much sometimes. Those of us that are more in the drug development field or investigating drug-drug interactions, we don't think about that so much. We want to talk about the impact the excipients have on transporters, specifically efflux transporters like PGP, BCRP, and uptake transporters like OETP2B1. I'm also going to talk about the impact excipients have on enzymes like the CYP3A4, CYP3A5, and excipients. I want to conclude by saying maybe that's an unexpected source of drug-drug interactions. Talk about drug-drug interactions. Well, drug interactions occur when a drug's activity is affected by consuming something else. Maybe that's a food, beverage, smoking, or other drugs. We're thinking about drug-drug interactions, but they can also be impacted by foods or beverages. And this could be due to competition for the same target in vivo. A common example of this, if somebody takes Ambien for sleep and has some alcohol along with that, well, both of those are targeting a GABA receptor. So you can get a drug interaction between the food and the alcohol that way. You can also get interactions because it's two things are targeting the same transporter or enzyme that are mediated that affect the drug's ADME properties, the absorption, metabolism, distribution, or elimination. If one drug is inhibiting an elimination pathway that's used by a second drug, you can change the pharmacokinetics of that drug. And one thing that's fairly obvious, but I put it here anyway, that the DDI potential increases with the number of medications taken. Polypharmacy, how common is this? One of the things we don't think about so much, how many drugs are people taking? This is kind of old data. I apologize for that. It's from 2010, but it's still relevant today, where the FDA looked at Americans that are ages 0 to 11, 12 to 19, 20 to 59, or 60 and over, and how many prescription medications each group is taking. If you look at relatively young people, it's not so many, but you still have people that are taking 2 or more prescription medications. Adults between 20 and 59, about 30% of this group are taking 2 or more medications, meaning there's a possibility of a drug-drug interaction in this group. If you look at people, Americans, 60 and over, 76% are using 2 or more prescription drugs. 64% are using 3 or more prescription drugs, and 37% are using 5 or more prescription drugs. And I want to say these are simply prescription drugs. This does not include over-the-counter drugs, NSAIDs, proton pump inhibitors, antihistamines. None of those are even included in this, and people take a lot of those medications. Nutritional supplements, the St. John's wort, whatever. All of those are drugs, and all of those are undercounted here. I haven't seen the complete data, but the CDC is actually compiling data from, it's 2022, but it's not released yet. These numbers are actually higher than they were 10 years ago. So this problem is simply increasing. One of the things I want to mention, a drug is actually more than an active pharmaceutical ingredient, and we don't think about that so much. Medications contain a variety of things that are called inert or inactive ingredients, which are the excipients. The US FDA defines the API as the active pharmaceutical ingredient, the compound that's intended to provide the effect you're going for. Anything else in that medication is inactive ingredients. These are the excipients, and they've defined this as any component other than the active ingredient in that medication. Now, every API might be combined with several different excipients in any given formulation. Why do we use excipients in the first place? Well, one of the chief reasons is to enhance the stability. You want to add something like a preservative or an antioxidant to try to keep that medication the way it's supposed to be, antimicrobials to prevent anything from growing in there, buffering agents to get it to the appropriate pH, sorbents to absorb any water that happens to be around. It could just be for something I call aesthetic purposes. You want that medication to have the appropriate color, flavor. You may need to add sweeteners to it, especially for pediatric medications, or coating materials. Sometimes it's simply added for manufacturing reasons, binders and adhesives and lubricants. This is in the manufacturing of these pills and tablets. You need to add materials to get them to adhere together and go to the right shape and whatnot, as well as plasticizers, solvents, thickening agents. We don't think about these things, but they are in the medications. One of the things, too, are simply bulking agents or fillers. If a medication, say 5 milligrams, like a 5 milligram tablet of simvastatin, there's simply not enough material there to form a decent medication, so more than 90% of what's in a tablet is simply fillers to make it big enough to actually be effective. Well, not effective, but big enough to actually be taken. And sometimes it's used to help improve the bioavailability of the active drug, and I'm going to come back to that in a minute. Well, one thing, we know that drug bioavailability can be influenced by transporters and enzymes in the GI tract. This is one of the reasons the regulatory agencies are always asking for, you're developing a new molecule, we want to know, is it a substrate or inhibitor of efflux transporters in the GI tract like PGP, BCRP, MRP2? Because those transporters can limit the bioavailability of numerous drugs. Uptake transporters like OATP2B1 that are present in the GI can help promote the absorption of some drugs. So OATP2B1 can take the drug and help it be transported into the enterocytes, where they can then make their way into the circulating blood. The efflux transporters, on the other hand, are pumping them. Once those drugs get into the enterocytes, the efflux transporters push them back into the intestinal lumen, limiting the absorption. Also present are enzymes like the CYP3A4 or CYP3A5 in the intestine, which can limit the bioavailability by metabolizing the drugs before they ever have the chance to be absorbed. So all these processes can be happening there. What about the excipients used in drug formulations? What I said was sometimes these are used to help improve the bioavailability of the active drug. Well, a little more than 20 years ago, I was working for a company called Xenoport, and what we did was we used transporters to help improve drug bioavailability. What we did was take something that had limited absorption and turn it into a substrate of active transport in the GI, transporters like MCT1, PEPT1, OATP2B1, to try to improve the bioavailability. It was a lot of work going back and forth between the biologists and the chemists, and we had some success. We got some drugs that were advancing into the clinic, and we were saying, good, we'll move on to the next step. And it got into the hands of formulation scientists. I didn't know anything about formulation. To me, formulation was, okay, take the medication, put in a pill, you're done. But apparently there's a lot more to it than that. So I was talking to some formulation scientists, and one in particular, she told me, what formulation is we add magic potions that improve drug bioavailability. Well, she obviously got her PhD at Hogwarts. I figured, I really like something, I like magic potions, I really do, but I like to know some sort of mechanism that's involved. And I was really wondering, could this be due to inhibition of efflux transporters or CYP enzymes in the GI tract? Is that how some of these magic potions are actually working? Well, it turns out that excipients that inhibit efflux transporters can improve the absorption of drugs. With the appropriate excipient, you block these efflux transporters, allow more of these medications to get into enterocytes and ultimately into the circulating blood, and it might be possible to achieve therapeutic levels of some medications at lower doses, which potentially could mean lower costs to the patients. This approach could also potentially be used, probably more relevant, for medications that just fail to achieve therapeutic levels due to intestinal efflux activity. Well, it turns out over the last 20 years, this has actually been utilized. There are several reports in the literature where you can look at drugs like etoposide, where you can add an excipient to this and see improved absorption of that medication. Topotecan has very poor bioavailability, but if you add excipients that inhibit BCRP in the GI, you can improve the bioavailability. Paclitaxel, another one where you add the appropriate excipients, you can improve not only the solubility, but the permeability, the bioavailability, and you can see these effects in vitro and in vivo. But one of the things here, excipients, while they can help improve the bioavailability of the target drug, they could also potentially impact the bioavailability of co-medications. And there might be some unexpected effects on other medications taken at the same time. One of the things I referred to before was all these excipients. It turns out that more than 70% of the mass of your average medication is excipients rather than active pharmaceutical ingredient. And if you look at the number of excipients that are in each pill, these are the number of pills, the number of excipients taken from these databases, we find that there's an average of 8.8 excipients in every medication. Some of these things contain well into the 20s, 20 different excipients that are in that same pill. And different formulations are produced by different manufacturers. When a company comes out with a generic medication, it has to pass a bioequivalence test showing that the effectiveness, the absorption of that is between 80 and 120% of the original medication, but it doesn't have to have the same excipients. Levothyroxine, one drug that's used commonly for thyroid deficiency, 43 different manufacturers produce 140 distinct formulations of this medication. There may be some differences. Drug-drug interactions. We spend a lot of time in my lab and other labs looking at transporter and enzyme-mediated drug-drug interactions on the active pharmaceutical ingredients. The regulatory agencies always request this information on APIs, and FDA, EMA, PMDA all have their regulatory guidance documents to help us understand what transporters, what enzymes need to be evaluated, what kinds of evaluation tests we should do. And the ICH is coming out with a harmonized guidance which should supersede all of these, and hopefully we will hear more about that this afternoon from Brian. So I'm not going to go into that in any detail. But remember, a drug is more than an active pharmaceutical ingredient. What if the other 70% of what's in that pill has an effect on transporters and enzymes? There are several reports in the literature that excipients can inhibit enzymes and transporters in the GI tract. Your CYP3A4, CYP3A5, CYP2Cs, efflux transporters like PGP, BCRP, MRP2, uptake transporters like OATP2B1 and 1A2 that are present in the GI tract. Excipients can have effects on all of those transporters and enzymes. There's a few reports, but most of those only look at a couple of excipients, and they use a wide variety of test systems. So it's very difficult to really compare between the different papers. Some of those papers are exploring excipients in Keiko cells or MDCK cells, HEK cells, other types of cells. Others use membrane vesicles prepared from SF9. Sometimes there are things like intestinal segments or gut sacs that are used, hepatocytes or microbes in these evaluations, or proteins. Some of them are in vivo, looking in rats. There are even in vivo studies looking in humans at these medications, or these excipients. And we wanted the ability to compare a large group of excipients in the same test system to get some meaningful comparisons between them. And the Bill and Melinda Gates Foundation gave us a nice grant to investigate the impact of excipients on the intestinal transporters and enzymes. So we were able to test more than 50, or nearly 50 of these common excipients as potential inhibitors of BCRP, PGP, OATP2B1, and CYP3A, 3A4, and 3A5. So thank you to the Gates Foundation. What we did was we looked at all of the excipients shown in this slide as inhibitors of the efflux transporters BCRP and PGP, the uptake transporter OATP2B1, and CYP3As, using a range of concentrations up to the maximum dosage allowed in oral formulations. And that information, some of it you can find in the FDA's excipient database, but not everything is there, because a lot of companies, their excipients become their magic potions. They're trademarked, and they do not disclose that information of exactly what concentrations are used. So I grouped these. This is kind of a random grouping of my own accord, but I grouped these to commercial excipients from the Abitec or Assay corporations, glycols and derivatives, things like polypropylene glycol, polyethylene glycol, some monolaurates derivatives of those, sorbates like polysorbate 80 and others, cyclodextrins, another very common one, beta-cyclodextrin, hydroxypropyl beta-cyclodextrin, surfactants, these are your detergents, bridge, Pluronic, other things that are used there. A group I just call natural products because I just didn't have a better way of describing them. Things like capsaicin, oleic acid, trimethylchitosan, and a couple of bile acids that are used in formulations. And we're going to show you a lot of data relatively quickly. The way all of these graphs are going to be shown is the activity on the left side, the concentration of the excipient on the x-axis. And if you see the curves where PGP in dark blue, BCRP in light blue are showing inhibition, these are ones where the excipient potentially is altering these and increasing the potential absorption of drugs in the GI tract. OATP2B1 will be in green. That's our uptake transporter. So if we inhibit PGP and BCRP and don't inhibit OATP2B1, we might see increased absorption of the medication. So these are the excipients from the Abitec Corporation. And what we can see is a lot of these do show inhibition of the efflux transporters and the uptake transporter. So OATP2B1 and PGP, we see pretty strong inhibition by Akonin C50 here. Akonin MC8, we see inhibition here. Granted, these are relatively high concentrations, 100 micrograms per mil, where we're seeing considerable inhibition. Capmol, another one of the Akonins, again, relatively high concentrations to get there, but ones that are theoretically possible. The ones from Assay, really not so much inhibition. The only one that did was this Gelusir 4414, where we see fairly strong, fairly potent inhibition by PGP and by OATP2B1. These are in the order of magnitude of about 10 micrograms per mil, and this can be provided in what's allowed is more than 1,000 micrograms per mil in the oral formulation. So these are concentrations we're seeing inhibition at 100-fold lower than what is allowed in that formulation. For the cyclodextrins, typically what we're seeing is inhibition of the uptake transporter OATP2B1 more so than the efflux transporters PGP and BCRP, as shown here for beta-cyclodextrin, derivatives of that, sulfabetadex. We're seeing inhibition of OATP2B1. We're also seeing some inhibition of the efflux transporters, but not quite as much for most of these. Polysorbates, another example where we're seeing inhibition of the OATP2B1 and inhibition of the efflux transporters. The PGP and BCRP a little bit less so than the OATP2B1, but we are still seeing inhibition there. And these again are relatively low concentrations, especially for OATP2B1. We're seeing the ones in the order of magnitude of 10 micrograms per mil, where we're potentially seeing concentrations that could impact the absorption of co-medications. Surfactants, detergents. We're looking here, again, we're seeing inhibition. It's kind of a mixed bag here. We're seeing sometimes PGP much more than BCRP with OATP2B1 kind of intermediary. And this is the bridge detergent. One of the things I will say is when you're using detergents, you can get membrane disruption. We're looking at PGP and BCRP in cell monolayers and measuring this using a bidirectional transport assay. But we also looked at this. We included a monolayer integrity marker to make sure we weren't at concentrations that were inhibiting by simply disrupting the monolayer. So we cut that off at those concentrations. Bile acids, we are seeing some inhibition of PGP and OATP2B1, a little bit more so than BCRP. The glycols, interesting group here. because we're seeing inhibition of both efflux transporters, BCRP and PGP, not really so much inhibition of OATP2B1. This was from polyethylene glycol 300. We also have propylene glycol, which is showing similar effects where we're seeing inhibition of both efflux transporters, less so for OATP2B1. And TPGS, vitamin E, we're seeing considerable inhibition of PGP and OATP2B1, not so much of BCRP. But this is concentrations that are definitely achievable in vivo. Some of the things from the natural products group, again, we're seeing inhibition mostly of the efflux transporters more than OATP2B1. Capsaicin was one where we see inhibition of both of our efflux transporters with very little inhibition of OATP2B1. Oleanolic acid, again, another one, we're seeing inhibition of both the efflux transporters. And glycerol monolinoate, also some inhibition there. How about the CYP3A? Well, excipients from every group also showed inhibition of the CYP3A, 3A4, 3A5. So we're seeing that with TPGS, bridge, polyoxyethylene 40-stearate, capsaicin, all the groups shown before. There was at least one example where we had inhibition of the CYP3As. This is a giant table that I know you can't read, but you can have a copy of my slides, where it compares the IC50 values, PGP, BCRP, OATP2B1, and CYP3A for all of the things that we tested here. I want to come back to a slide I showed toward the beginning. How many excipients are in all those drugs? We know a lot of people are taking 2, 3, 4, 5 or more medications. Every one of those contains excipients, and the average number of those is 8.8. So what happens for somebody that's taking 5 or more medications? How many different excipients are they getting? And even if the FDA database allows, a milligram, 10 milligrams, whatever it is of this excipient, what if it's in every one of those medications that that patient is taking? Well, we use in vitro assays to predict the risk of DDIs, generally to support IND filings. And we do that by testing the active pharmaceutical ingredient as a potential inhibitor and a potential substrate of PGP, BCRP, a variety of uptake transporters. But one of the questions that's left unanswered, what if the drug itself isn't inhibiting, but the excipient used in that formulation is inhibiting BCRP and PGP? And what if that's happening to somebody who's taking a variety of medications? Say a patient who's taking comeds like digoxin, which is responsible for increasing blood pressure, and dabigatran, which is an anticoagulant. These are drugs that have relatively narrow therapeutic windows, and if you're allowing a lot more of these drugs to be absorbed because you have excipients blocking these transporters, that may not be a good situation for that patient. So could that drug's excipient, rather than the drug itself, cause some adverse events, resulting in a clinical DDI when the in vitro test failed to predict one? One of the things I should have included in this talk too was the regulatory agencies really focus on cutoff values to avoid false negatives. So there's always a safety window built in. They're looking at 10 times the IC50 or KI value, 50 times the unbound. But we still have false negatives, and those false negatives are unexplained. Maybe that's due to some of these excipients that are used. Just one possibility. Another thing I sort of alluded to before, the innovative versus generic medications. Well, when a company comes out with a generic medication, it has to pass the bioequivalency test. But drugs from different manufacturers may contain different excipients. That drug by itself may be equivalent bioavailability, but it may be bioequivalent, but it might have different excipients, which could impact co-medications. Of the 18 most frequently prescribed oral medications in the US, the number of alternative formulations per API is 82. That's an average. Levothyroxine had 140. That was the most extreme example I could find. But that's a lot of different formulations. I know I have a couple of prescriptions that I get, and every time I go, it seems like some generic manufacturers come out with a new version that's a little bit different. So the manufacturer keeps changing every time I pick up my drugs. It's like, okay, I'm sure they're bioequivalent, but I'm sure they also contain different excipients. And the question is, could you get some clinical DDIs resulting from some formulations but not others because they're different excipients? I don't have the answer to that one. But I want to wrap up by saying many pharmaceutical excipients can inhibit the GI uptake and efflux transporters as well as the CYP3A enzymes. Inhibition of these GI transporters and enzymes can increase the drug absorption of both the branded, the formulated, innovative drug, as well as co-medications. And the choice of excipients and drug formulations could really significantly alter the pharmacokinetics, pharmacodynamics, and bioavailability of drugs, as well as co-medications that the patient is taking. And it's at least possible that excipients could be an unexpected source of DDIs that the in vitro test that we conducted failed to predict because we only looked at the API, we didn't look at the excipients that came along with it. I want to wrap up by thanking the people that were responsible for this. I didn't run any of these experiments myself. This was all the work of Megan Graves and Anna Rodriguez, Yue Du, Eileen Kaufman, Ron Latham, all involved in running the assays. Rob Taylor, who was responsible for putting together this collaboration with the Bill and Melinda Gates Foundation that allowed us to do this work. Collaborators from the Gates Foundation, University of Washington, Joe Pauly, and the suppliers of the excipients. And with that, I know I have time to take a couple of questions.